Okay. Hi, everybody. Welcome to the biodiversity seminar. Today is a great pleasure to have Carlos. Will give a talk about diversification, threat architecture, and coexistence in complex ecosystems. Actually, I don't know Carlos in person, but I am very happy to be here because he was one of the first authors who bring me to this field of network ecology. And I had the pleasure to read when I started my bachelor thesis on the topic. So it's a great pleasure to listen to you, and the floor is yours.
Thank you, François, for the invitation. Thank you, everyone, for being here. And also to the people online today. Things are becoming more and more sophisticated. We are trending towards a hybrid system, which is quite interesting from the speciation diversification point of view. Just a quick kind of overview before I start. This is a talk that has been growing the last 2 years. When I started to shift from, let's say, food webs and ecological networks to still working in networks, more from a diversification point of view. And I think this talk has been consolidating, and maybe today's version is the most unstable version from the last 2 years. I don't know why, but so I would try to be really compact and specific, right, with the talk, but it contains a lot of fluctuations. It's a bit of a Tarantino movie. It contains different parts, so you can interrupt me anytime, right? If you feel, hey, where are you going, or can I ask you a question? That would be great. So this is a teamwork. So I'm using, it's interesting, just quickly, a technology that allows you to go, it's a matrix. You go down, you go right, you go backwards, you go up. But these devices contain only the row, right? You go in one line. That's interesting. So these things are going to be evolving soon towards this matrix format of presentations. And so this is a teamwork, more than 10 years. I would mention here Cecilia on top, also Francisco Encinas, Ali Badati, Jorge Rodriguez, Catalina Chaparro, Paolo Guimaraes. Many people have been contributing to all the ideas and methods and results I'm going to present today. So it's a team bottom-up work. And this you can download the talk, right, from my GitHub, established. And so I would like to introduce briefly the questions. What is a species? It seems a trivial question, but for me it's the most difficult question in biology. Then I think a common question in biogeography, that's diversification increase in the tropics. It's an old question with new results, new patterns emerging. Then why are there some with many species while others have just a few? So the first part of the talk is more external factors driving diversification. Then there is going to be a shift towards internal factors like genome evolution, development, and then at some point I will try to join these two parts to see how the future is going to be. And then a synthesis for a diversification theory. You can download the talk, and there are many references there. It's not an exhaustive list, but it can maybe drive you through what is a species, right? It's minor research, but sometimes interesting, right? To guide if you want to go deeper. Then the lineages, right? Diversification and the coexistence and the unifying diversification. So what is a species? We have been thinking a lot, and many people, right, in biology have been thinking a lot in defining a species concept. But technically, this was, I think, has been my main frustration as a maybe conceptual definition, like a subset of compatible, right, from the reproductive point of view, compatible individuals is the standard. There are many other potential definitions. I think they are all valid depending on your questions and your problems. But then technically, it took me years to understand the concept. And I'm going to try to introduce you here briefly one of the ways people have been modeling the species concept. There are many others, but this, I think, is from the quantitative terms, is one of the most accurate. You can tell me, no, you are wrong, Carlos. I think there are many others more accurate, and then we can open the discussion. But one of the problems for defining a species is the species boundary, how we define a boundary. And there are potentially many boundaries, and which traits define a species, because individuals contain many traits, and variation is pretty large in populations. And compatibility in reproductive traits is also a bit tricky, because not all the individuals in a given population are compatible from a reproductive point of view. So the basic definition still is a bit tricky, because they belong to the same population, to the same potential species. So defining the boundaries in this animation, you see, like in time, the productive trait is expanding the variance. So, you know, you have some mating schemes, some processes, and instead of the variance stabilizing with the reproductive traits, it's expanding. It's one of the potential outcomes. There are many, many outcomes. So let me introduce you the quantitative definition of a species in a toy model. So imagine we have a population of individuals, J, is rows of 1, 2, 3, 4, K individuals, and each individual contains a genome size L, very large. Each dot there, each circle is a locus, right? It can be, you in your mind, that is the genome in there, right? But you can define it at many levels. But here you have a pretty large genome for each individual, and each site contains a mutation rate. And then with these elements, these basic elements, you can put this population in a mating graph. And you can study the network properties as we do in ecological networks. And a mating graph essentially is going to tell you here that the lowercase qij, that is the connection between two individuals. Right? If they are compatible, you put a connection. If they are not compatible, you remove the connection. That's it. And so you have a threshold, which is a parameter q mean, you see there, right? That defines, right, the threshold if two individuals can reproduce, can have viable offspring or not. This is the standard definition. And so this matrix can have many points, many sets. In the graph you see here, you see the black lines, these are the compatible individuals. And all the underlying lines are incompatible individuals. So you can start with this graph and think about what are the conditions to have a species following the standard definition of a species, right? A subset of individuals that are compatible in reproductive terms. So the cartoon here, you have imagine many scenarios. Individuals arriving to a place, a lake, a mountain, whatever, is a subset of individuals, and now they form a family, and they are most of them compatible. And then at some point they start to diverge. Some individuals start to mate in the corner of the graph, and then they start to diverge step by step. You can think here in generations. Only mutation is happening, right? It's a toy model. It's quite simple. And then at some point, the variants start to emerge till you get isolated clusters. This is quite intuitive, right? For many of the problems we deal with in biodiversity. The point is how to model these things. For this definition, we use an individual-based stochastic model with birth, deaths. That's it. It's an isolated network. Right? No spatial structure. It's a simple model. But for this network, there is a solution. For a stochastic process of birth and deaths, as a demography, there is a solution. And essentially, it tells you that if the parameters you start with, J, total population size, mutation rate, and Q mean, the threshold to define compatible individuals. And if the Q mean is larger than the mean of the similarity matrix at a steady state, then you don't have clusters. You have a mean value with a fully connected graph, or a graph that has been losing connections, but you have only one species, no speciation events in that graph. But if the initial conditions, especially the Q mean at the steady state, right, is lower than the mean genetic similarity of the population, then you have the formation of clusters. You have a speciation, you have a species forming, a subset of individuals that are, you can reach all the individuals in that subset via connections, and you are isolated from other clusters in the graph. So it's the standard definition of a species. This can be extended to sexual and asexual populations. Can be extended in many, many ways. Can be by using the combination. You can use even complex states, meaning pleiotropy, epistasis, many genomic driven processes, which is, I think, quite interesting for the next part of the talk. But for populations that reproduce as clonal, asexual reproduction, on your left, you see that you have a node on top, individual A, and then you have individual A1, A2, A3, AN. And then you have the continuous line is that they, right, you have mutations, but they essentially are producing clones. And they are at the genetic similarity threshold applies for the two cases, they still can have horizontal transfer, right, in asexual population. And in the sexual, they do mating. The continuous they can mate, but at some point, you can derive for this toy model the number of steps you need to, right, given the mutation rate, total population size, and the threshold, the minimum genetic threshold to have compatible individuals to produce an species, to produce an speciation event. So you can calculate the number. And essentially, for asexual reproduction, the number of steps is given by the log of the Q mean, the threshold in the initial parameter, divided by 2 times the mutation rate, which is quite interesting, right? From the theoretical point of view, we love this, that you can derive analytical derivations given a complex graph. And for sexual reproduction, it's a bit more sophisticated because you are mixing individuals. And so the question becomes a bit more tricky. And if you are hybrids, meaning when you have clusters, you start to combine the clusters, the model is not, you cannot do analytics, you cannot do calculations, you only do approximations, simulations. And so for this specific toy model, the number of steps to get an speciation event is larger in the sexual, is n star asex, larger than n star sex, meaning you need more generations to have an speciation event in asexual than in sexual populations, which is something intuitive because you are mixing with sexual populations, and then things are faster somehow for this toy model. And essentially, when you divide one divided by number of steps, you have the speciation rate. You have the number of species that are given this model, this toy model, can produce for sexual and asexual populations. This is kind of the first part I want to introduce you, how we deal with the species definition from the theoretical point of view. This follows the standard definition of a species. But let me go a jumping process here. So we go to a question that I think most of you, I'm sure, you have been working in biography, no more than me. But to build a theory, specifically in diversification theory, we need to touch many fields you are not up to date. That's a bit frustrating for building a theory. But let me, like surfing a bit in these ideas. So that's interesting because I think this is one of the first concepts you get when you start to study biodiversity. This is one of the most fundamental, right? You say, well, today, right, there is a latitudinal, and the point is, okay, does this gradient work always, right, in deep time, or has been fluctuating many times? So today is just context dependent. I mean, I'm sure you have a much better maybe understanding, right, from the literature, new results. But for me, this is an interesting, right, because it's around the idea of external factors driven diversification. And there are many extensions, right, mostly coming from plate tectonics, right, from landscape dynamics. And there are many models, right, simulation models, making clear that landscape dynamics is a strong force in driving diversification. And I think this is quite a standard today. I would say that the diversification theory coming from external factors has been evolving faster than the one coming from internal factors. from genome evolution, development, and ontogeny. But for this external kind of elements to produce diversification, if you go backwards in time, it seems that for some taxa, it has, I mean, it didn't happen. So essentially, it's mostly because landscape masses and other potential factors have been evolving, because mostly continental drift, but landscape dynamics, you can put here many factors. And then for many paleo data, the gradient is not there. So if we go deep in time, it seems, right, that the pattern we see today, even the most general pattern in ecology and evolution, is just specific for contemporary time in the history of Earth. So now, yeah, sorry. I love this. It's a jump, but I really had to tell you, I mean, it's something all these guys have been working for many years, putting data together to understand plate tectonics. I think that's something I miss in biodiversity. This is a constructive kind of critique. So how many people need to work together to really have a much better understanding on diversification, right? In the history of life on Earth. We maybe are not there yet, but many different people in many different disciplines, they have been putting data together, and they have been assembling such a beautiful, right, multi-data sets to really have a compact, coherent view, in this case of plate tectonics or global paleogeography. So many people here, WSL, Fabienne, Loic, many others, they have been working, contributing a lot to this theory, the diversification theory from the landscape or seascape dynamics point of view. So in this case, you see the right side is I don't remember, is the model the right side? I think, yes, the right side is the prediction from the model, and the left side is for color. And there are many taxa and different groups analyzing in this study. But essentially what you see is in time, the hot spots in green have been moving from the Tethys Sea, the today Mediterranean Sea, towards the Pacific Ocean. And many groups follow essentially the seascape dynamics through time and space. And this is pretty interesting because you don't need to put many factors, right, underlying diversification to predict at least the spatio-temporal trends on where the hot spots have been going in time and space. That's pretty exciting. As I told you before, there are many papers on this line, on this topic, and the progress is pretty amazing. And really, the progress of external factors predicting lineage diversification. However, I need to transition. I need to transition towards the internal factors, where I think things are, I don't know why, maybe it's because biology is so complex and the levels, right, and only an organism are so large number of levels that the progress is a bit more complicated. But we need to take the risk. We need to take the risk in trying to understand if the external drivers and the internal drivers, when we put together these two drivers, we understand, we build a more solid diversification theory. So the summary here is, okay, the latitudinal biodiversity gradient in deep time. So environmental gradients and landscape dynamics in time and space are external explanation for diversifying needs. This is quite standard. I'm really happy that the progress in this field has been so fast. And now comes the tricky things, right? Because if you go to more detail, you collect data different levels, phenotypes, genotypes, then you start to see the variations and the complications. One is the cyclic radiations in Africa. So if you go to the details here, in blue, these are lakes where some of the radiations for cyclics, they never happen, even with the same conditions. Right? The lakes, they have similar conditions in light, in nutrients, in many other factors, But in some lakes, you see radiations, and in other lakes, you don't see them. The question is why? And they are all in the tropics. So, and some radiations are huge, 200 species in less than 10,000 years. Other radiations are small, 5, 10, 12 species. But the question is why? So this is something, right? One explanation is, okay, landscape dynamics again comes here at a more micro meso scale, right? Within, in a network of lakes, in dendritic networks, right? And maybe this is an important component to produce such a variation in a given setting, in a given context. However, I'm trying to enter in a more stable part, right, of the discussion. So if you go to experimental evolution, it's many old results, right? You put bacteria in some, in some nutrients, and then you let them evolve. They start to form clusters in 2 and in 3D. New types emerge, new phenotypes, new genotypes. And this is the classical standard, right? Diversification by niche-driven, right? Diversification in a few generations. And so this is beautiful, right? Because you say, well, speciation can also happen in sympatry, right? And when you put these things in the context of the external factors, then you start to think, well, but the speciation can happen in sympatry, or is all allopatry or parapathic speciation, where you have a combination of the two. So this is then when things become a bit more tricky, because if a speciation can be in sympatry, this means that essentially the populations, the behavior, the phenotypes, depending on the mating schemes and many other elements, right, can diverge locally and can produce a speciation even without landscape dynamics. And this is something that maybe most of you already experience when exploring biodiversity. You go to conferences, biogeography or ecology, this is completely missing. If you go to conferences, evolution, the other part is missing. This is essentially selection. So each one goes to where you prefer, some preference. That's great because it's part of the limitations we have. But if you want to put things together, you need to be in different settings. You need some exposure to different views. And the point then is how to build a theory of how to build databases and how to improve our understanding of nature and diversification and biodiversity. So the point here is, what if the DNA, right, genome processes, development, gene expression, behavior, sexual selection? This is not me. This is not me. This is because I have been trying to learn from many people from different fields. And there are many, many results showing that these elements are important. So should I stay in the same place, right, doing the same, or should you, confront reality, which is more complex? And so are all these things embedded in complex trait architectures combined with external factors to produce a set of interacting layers, challenging any attempt to build a solid diversification theory? This is crazy, right? Because at some point you say, well, I said fabulous, right? Because it's too much. But I think for many people working in biodiversity theory or diversification theory for many years, we have a similar feeling, right? The point is how brave now you are, or how naive you need to be to really move forward a synthesis in diversification. This is the main message of the talk, right? I think we are not there. But just as a starting path, right, towards this synthesis, if you read about systems biology, the evolution of modularity is there. It's one of the mainstream concepts. Essentially, the idea is you have genotypes, right, black dots. You have phenotypes, white dots or cycles. And then during development, you have the genotype-phenotype map for a given trait, but for many traits. And for each individual variety, right, during ontogeny, during development. Unfortunately, because you can say, well, we can skip all these things and keep things easy and as toy models, which is great sometimes, as the species definition. But things are a bit more tricky. And now the point is, you can have an integrated phenotype, meaning that essentially you have large parts of the genome correlated with the phenome. They are highly integrated. Why? Many potential reasons. But you can go from that boundary to the boundary where the phenotypes are modular. You have a specific parts of the genome or the phenome. As you see here, the phenome is a network. You can have the phenome, right, interacting without the effect of the genome, plasticity and many other potential mechanisms there. But the point I'm making here is that you have a gradient from fully correlated, integrated phenotypes to fully modular. So why I'm coming here? Because these can be connected potentially with the classics in biogeography, right? If you go to the classics, right, the Grinnellian niche, the abiotic factors, there are many, many papers, right, showing that the abiotic factors are really important, right, for niche modeling to predict distributions. But then the Daltonian niche comes with the biotic interactions. And these are also important components, right, in predicting distributions of a species. But then Hutchinson came and said, well, that things should be connected somehow. And then in contemporary time, we had the dispersal, right, the limitations, and many traits associated with dispersal and migration. So this is the BAM, right, the BAM, the BAM model, is classic in biogeography. But now the point is, this is still all kind of disconnected. The point is how we connect these things. One way, there are many, is okay, you go to the data, you go to morphology, to reproduction, territory, physiology, phenology, behavior, many, many, many data from different individuals belonging to the same group somehow, and you can put together like the BAM, right? The abiotic, the biotic, and migration traits, for example. And on top, you have one cartoon saying, okay, for these three traits, they are highly correlated. This is a cartoon, right? This means that essentially the biotic traits and the abiotic traits are correlated. If you respond to the biotic, you are at the same time responding to the abiotic. Sometimes they can enter in conflict. Right? If you decouple them somehow, you respond independently. But if you couple them, positive or negative, then things are a bit more tricky. You have some constraints. And with modularity, essentially, is that you respond independently. These are two scenarios. And the point now is how to model, how to build a framework where you can put these things right together with fitness demography to understand how species right diversify and reproduce the empirical patterns. There's still nothing about spatial right context or landscape dynamics. Right, this is, but I am now going to the internal mechanisms, right, to produce divergence. One framework, there are many, 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 and we have been working this for years, still not finished. But one way to do this is you have three big columns here. You have the left one, genes to phenotypes, the center is phenotypes to fitness, the right fitness to biodiversity. Each one contains many fields. So we need some simplification, right? These are brutal simplifications, as we usually do in science. So the first column essentially is telling you trait architecture, right? You can have the BAM that is modular. You see the BAM, the genes to the traits is modular. Right? You have a specific genes blocks producing, expressing some specific traits, and this modularity can be, like independent traits. And when you go to integrated, you start to have correlations. This is the first element, but this element contains underlying mechanisms. And three of the key mechanisms coming from population genetics are pleiotropy, which is essentially a gene to trait matrix. It's a binary matrix and essentially contains, right, who is interacting, right, with specific traits. So you have on top, right, gene to trait in pleiotropy matrix, in the middle epistasis, which is essentially the quantitative effect, right, that genes have on specific traits, and then the expression vector, which is essentially changing during development, and also is quite context dependent for many, many organisms. But these three elements can be modeled. You can put them in numbers, and then you can go to the central column where you see selection, and here we enter in the many ways to model selection via fitness. There are many potential selection modes, right? You have stabilizing selection, essentially an optimum, and all individuals track that optimum. The optimum is static, and all individual traits, let's say, they are biotic. They follow the optimum, and then the individuals that differ from the optimum start to die with higher probability. This is a standard way. There are many others. You can also say that the optimum is moving, and then individuals are tracking the optimum. You can also say there is no optimum because the individuals have a tolerance that essentially is beyond, right, in a given environment. So there are many ways of modeling selection. And the point of this framework is that we have these many ways. We have a population of selection modes. And essentially, you have the three distributions. You have for the biotic on top, the prey and the predators. You have the abiotic via optimum, and then you have the dispersal optimum. It's just a cartoon. It's just to show that you can connect, right, genomic processes, the genotype to phenotype map, to selection and fitness processes, and then it comes to demography, to population dynamics. And population dynamics essentially is if you have a spatial network, then you can compute many metrics. Right? You can compute intraspecific genetic or trait variation, or intra versus inter. Right? You can have coexistence metrics. You can have functional metrics. You can have many properties calculated at the local scale, and you can also have a metrics at the regional or a global scale. The point is that you need a framework to understand the coupling and the decoupling of these levels, to understand the emergence of biodiversity, and also the conditions to have diversification. This is spending a lot of time because it's something that you ask yourself, is this worth? Is this building a framework really needed in science? I'm a bit neutral, even if I spend a lot of time on this, but this is an interesting question by itself to discuss in science. There is a large cost to do frameworks today. This is my bias here, right? I'm neutral in being naive, in trying to understand nature, but then at some point you realize that this is a huge cost in building a framework. And essentially, it may be similar to the idea of a cost of hybrids. And this is also open to discussion. So to finish the talk today, I think we have been doing simulations with this framework. There's two scenarios, right? The modular and the integrated. We call here, right, modular and magic. Magic is integrated. I like the word magic is when you collapse everything to one thing, which is great. It's essentially when all is correlated or is integrated. And so we explored two gradients: migration gradient in a spatial network from x-axis, from minimum to max, and then the biotic selection gradient, meaning how strong, right, the interaction with your predators or prey is, right, for the population dynamics, for the demography. This is a context of stabilizing selection. So it's one specific scenario. You can have many outcomes depending on how you obtain the selection and the fitness functions. But for this scenario, the color bar is the species frequency, is a biodiversity metrics. And what the general scenario is a bit blurry, but the general scenario emerges is essentially that when you decouple the evolution of modularity, essentially is increasing for a long gradient of migration and biotic strength and interaction strength, increases the number of species. Essentially, the higher the modularity for any species in the system, the higher the probability to persist. Meaning that any type of constraint, any type of increasing correlations, positive or negative, between these B, A, and M traits, so biotic, abiotic, and migration traits, the lower the number of species. Many things here, right, to discuss. For example, you can ask, well, but where are the nucleus here? This is a neutral landscape, meaning that in principle, right, is quite, even if you have biotic interactions and migration dynamics, but the landscape is static, the environment is not fluctuating, you don't have strong gradients. There are many assumptions here. and things might change. But I want to illustrate here that a framework to understand diversification from genomes, right, to biodiversity is possible. It's still quite challenging. So the last slide is for me one of the, let's say, most complex slides to understand diversification from the internal point of view. And is a work by David, Joanna, and Ole. They have been working for years, right, in sympatric speciation, but is essentially a way to contrast with external factors, right? There has been also a lot of progress in genome evolution to understand diversification, and maybe this summarizes, right, many of the aspects. Essentially, if we go through, right, speciation or diversification contain many stages. I'm mentioning this, or I am, like bringing you here because when you go to the external factors driven diversification, most models assume that the speciation is easy. It's just a mutation. But for people working in speciation, it's a nightmare. And this is the problem in surfing, right? In surfing with different people, with different mindsets and different data sets. So the ideal is to put this in a complex landscape, right? In complex dynamic landscapes. It's still not there. But diversification contains steps. Divergence in the top is absent because population is in the same place, a few generations. Then, some mutations happen here and there. Allopathic speciation, you see, there is a barrier and you have two populations. But then with a non-allopathic is you have still some flow, gene flow, because some populations, they connect each other, they migrate. So this is quite intuitive for most of you. But things are becoming a bit more tricky because the sources of genetic variation can be many, and also the mechanisms, right, start to be a bit more tricky, like the genome, right, driven mechanism, pleiotropy, epistasis, many recombination factors, but the mating schemes, behavioral schemes, so things are quite tricky at that level. So you start to have divergence, right? Do different paths in the diversification, and then we enter in intermediate stages where the third row there, you have combinatorial mechanisms. You have potentially new factors coming into play to predict divergence in populations. So that's something interesting. I think that is one idea. I don't want to go to the details here, but it's one idea I think we all experience is why we go to the detail only in things we are interested in. Why I don't understand. Right? We should go to detail to all the things, right? But it's impossible, maybe for human mind. It's the reason we need things to complement each other, and each individual can go to a detail of a specific problem, and all of them together can go to all the details. Another problem is how to put all the details into a framework. Is the sacrifice part, right? You need to sacrifice some elements. But I think it's the main idea. It's maybe a trivial idea, but when doing synthesis, it's essential. It's essential to explore this trade-off. So just to summarize the take-home message, speciation is a complex process. I mean, physical, chemical, biological processes drive the divergence of new phenotype maps and complex trait architectures along a sequence of diversification stages. External and internal factors combine to produce diversification patterns in space and time. They are highly heterogeneous. And a diversification theory connected to is currently missing. And the last kind of take-home message, toy models are useful to understand basic diversification processes because many reasons. I saw you the first one, the species concept. It's a toy model, but it captures some elements of what a species is. Current data suggests that such systems biology, genomics, genomes, fluctuating environments, and landscape altogether are needed to connect micro to macro diversification and biodiversity patterns. Thank you. Yeah, Catherine. Yes.
The magic and the non-magic. I can't remember what it was. Sorry,
The magic.
the modular and the magic. Oh yeah, that's right. And
Yes.
so that modular and magic is the output of this framework you're trying to put together. Can you just explain that link
That's,
a little
very interesting.
bit more?
What is your
I feel like
intuition?
I've
What is
heard
your
of-
what is your intuition?
I mean, I think I've seen this before a little bit. Yes, I think your attempt is to take this framework and all these processes and then create this.
But the magic, imagine an individual of a long lifespan that is having fluctuating from modular to magic. This can happen. But in here, right, individual that is magic for the whole ontology. An individual that is modular for the whole ontology. It's an assumption. But this, you have a trait architecture that is fixed.
But then when you look at those graphs,
Yes.
you don't see much pattern.
Which one? Here. Yes, for that scenario, you don't see, you see a general pattern, which is, right? You see that the modular is higher.
Yeah, okay.
Overall, the gradient, along the gradient somehow.
Hmm.
That's it.
But within magic and modular, you don't see.
And we are trying to understand why.
Okay.
Yes. But one point here, I think you are bringing this point, is that the trade architecture is fixed. But from some data, from some taxa, trade architecture is dynamic during development, meaning you can shift from modular, right, to correlated and back.
Mhm.
And reasons can be plasticity. You have a stress, right, environment forcing you to correlate two traits in response to specific situations. But then, as soon as this is gone, these two traits respond independently. This have been observed in many data sets. So, but here we are not accounting for that. In behavioral traits, it's similar. Some behavioral traits sometimes are correlated, and other times they are working in a modular fashion. So maybe your question maybe go in that direction is more understanding this pattern.
I think I was just more simply trying to understand
This pattern. Yeah.
I have one also about these simulations. Like I'm not sure I got it well, but do you start with a fixed fixed number of genotypes or patterns, and then you make them evolve, and then your measure is a measure of persistence that you make?
Exactly.
Okay.
Exactly.
So you start like with a given number of genotypes or like
exactly. Okay. The starting, let's say you start with the simplest scenario, right, which is randomly generated, like trait architectures or genotypes, and then you start to have the mechanism described here, right, the pleiotropy pieces acting on the new offspring.
but the genotypes evolve.
The genotypes evolve. You have mutations. Yes, you have mutations, and these mutations are changing, right? These matrices and are evolving essentially. The traits are evolving.
And can the species? I mean, can they diversify? So can you like shift from, for example, one genotype at the beginning can become two?
You can diversify. Okay. Yes, you start with one species,
and then you
diversify.
Just because it's on this topic still. So then you sort of made this argument about plate tectonics and all this stuff being the, this really important biogeographic, thing and all this new data, et cetera. So I guess you have this to some extent with your movement trait, but that's maybe not necessarily capturing these sort of biogeographic patterns of connectivity, unless you're somehow giving all the species the same movement rule for some period of time in your simulation, assuming that these continents or these places are somehow connected. So is there a way to get, I assume this is not a spatial model. Maybe I'm wrong.
This is spatial. It's spatial explicit, yes.
A 3D, like a 2D.
2D. For now, it's 2D.
Sorry,
2D.
2D.
Yes,
meant
a 2D. You have a 2D landscape.
So is the 2D landscape then considering these biogeographic patterns of continental drift or whichever patterns we think are important as you set out?
Well, that's a great. The point is, let's say you have a biotic trait responding to to an optimum. This is more or less your question, right? You move in a space and you have a gradient, and depending on where you are, then your fitness emerges. If you are close to the optimum, your fitness is larger for the abiotic trait. This is, and you can turn off the biotic and the migration trait. Turning off meaning they don't have effect, for example, on fitness. So the idea of trait architecture is essentially the same idea than when you have one trait, but now you have many traits. And the point is how you count the fitness for the contribution, of fitness for any trait. So I think this follow your question. It's a biogeography model, essentially. But you put genomics, The things come from the genotype-phenotype map. So the individuals are not just, an individual with a given trait value, but the trait value is calculated from the genomic on the genotype-phenotype map processes. But it's a biogeography model, essentially, in static landscapes. Meaning the landscape is not, is not changing, or the environment is not fluctuating. This, you can add these complications, but at this stage, it's a static landscape. Say it again. Well, this is a Julia package. It's called Evodynamics. And we have a number of individuals, generation versus time, You see number of individuals, generations versus time, plots, and these are linear increase in computation time. With Julia, with R, we did a first test, it doesn't work. But with Julia, we can reach 1 million individuals, 1 million, 10,000 generations in a few days. We are on these numbers, but this is, if you take a look at the frameworks somehow, this is the main concept. Yes. Yes. Exactly. Yes. For example, 10 million individuals, 10 million. This is months. And yeah, 100 million individuals is years. Yes, so it's quite, but 10 million, yeah. Yes, and this is something maybe interesting, at least for people interested in building frameworks, is that you have some parts that can be calculated analytically, and then you plug in the parts you cannot do the numbers, and then you speed up things. This, for example, can happen, but as always, is at the cost of simplification, If you assume that, for example, that's interesting, The evolution of recombination. Recombination can be fixed, but can be also evolving. So the clusters that are forming at the genomic level are evolving, are changing the size. And so when you put a fixed recombination rate, you can derive analytically many things. But when the recombination rate fluctuates, then things are a bit more tricky. But it's the same for landscape dynamics, When you have static landscapes, you can do numbers. When the landscape changes, let's say the sites expand or contract, or the connections, And you can correlate, say, the sites contract, and then this site becomes isolated, for example. And if the site expands, then it starts to be connected to other sites. You can put the numbers around this, but now deriving analytically solutions is a bit more tricky. The same happened with so one general thing here is the more dynamics you put at each of the levels, the more maybe realism but the more complicated is to derive analytically some solutions. This is maybe a bit tricky, but yeah.
I have a question in the same direction of what you're discussing right now. It's related to how do you see this integration between these two isolated universes by geography and these genetic explicit models, especially because of the challenges related not only to the computational challenges that we have, but also there are some conceptual challenges that some processes that are assumed as processes at biogeography level are not processes at a more finer level of organization. They are just emergent properties from a finer level of organization. So how do you see this type of integration?
Great question. How do you have an idea?
No.
But let's see that a naive guy comes, right? A naive meaning here working neutral theory. And let's say that you develop a neutral model without emergence, right? No emergence. A neutral model. Most people would say, well, it is not realistic. But as a scientist, I feel it's good to debunk your own prejudices somehow. Why should emergence exist? Maybe it doesn't. The point I'm making here is you need to go to the simplest neutral toy and say, okay, can I predict the empirical observations? This is going in your direction. And then if you don't predict these emergent properties coming to different levels, then you need to take into account elements at that level. But this comes later, I would say. This is my way of proceeding. Maybe, and the reason for this is first, neutral, minimizing bias. Second, mathematically you can do numbers. And third, why assuming complexity when simpler things can explain the data at different levels? This framework looks complicated, but if I ask, for example, you in the field you are working, it's extremely simplified. Because when you go to different levels, you need to simplify the different levels to put them together. This is again the idea, you cannot go to the infinite in each level. So this is the trade-off, right? So, but I think the procedure is more or less clear, right? It's neutral, easy, no emerging properties, and test with the data. If you don't predict the empirical pattern, then you go to another level when you explicitly add these properties or the properties emerging at each new level to see how close you are to the empirical patterns. That's this more or less, but it's complex. I agree, it's quite complex.
Are there other questions?
You mentioned that how you implement selection can considerably change the outcome of this model, if it's like stabilizing or not. Did you, maybe I missed this, did you already tested for the outputs of the models under different selective setups? And do you see the same difference in species richness between the modular and the magic scenarios?
Yeah, that's a great, we are testing, these are all preliminary results. We are testing different scenarios. The first, probably what we discuss now, is the neutral landscapes, meaning, right, for this framework, neutral is relatively easy, right? Fitness is equal for all individuals for the 3 traits during all the computation time, and then you can derive some of the properties at the species, at genotype, phenotypic, and species level. But then comes different selection modes. This is the tricky part. Stabilizing is relatively easy because I think the stronger the stabilizing selection mechanisms, the faster you go to the mean type, as expected. So the variants are gone, and then you can obtain the solution at the infinite limit somehow. We are working on this. These simulations are right following essentially the infinite limit because you know the model is stabilizing selection. Now comes a bit more tricky. It's frequency dependent selection. which is quite complex because essentially what we are saying in frequency-dependent selection is the fitness function is telling you that locally you increase fitness if you are rare. For example, a resource, a prey that has a fitness, a trait value that is quite far from the mean predator, you increase fitness. And the common prey type, decrease fitness. So essentially, preys are diversifying locally by frequency-dependent selection. And the point is that this is a bit more tricky because you have to put boundaries to the variation, right, locally. We are exploring these things. The problem is we don't have any analytical expectation for this. And so you have some proxies. If you go to the speciation theory, the brute force speciation theory, brute force meaning the complex models that emerged in the 80s, these are models with 20, 30, 40 parameters. And there is a battery of models in speciation theory. Population genetics, I would say now this is maybe a bit not humble, right, claim, but so population genetics advanced a lot in mathematical terms, and then the speciation theory followed the population genetics. So the models became really complicated, but many models have been dealing with frequency-dependent selection to predict the speciation events. It's essentially right using adaptive dynamics. So adaptive dynamics here is not the speciation concept I put in the beginning, but there are many mathematical formulas to obtain when a species emerges, given frequency-dependent selection. Right? Where rare variants, when they can invade, you can calculate, the derivatives, and you can obtain the new types forming in a given local setting. What I'm trying to go here is the framework. You have the skeleton, but then the methods you use depend on the selection type. This is, I don't know if this clarifies a bit.
I think it's noon, so this is usually when we wrap these up.
Yes.
And Nadia, do you want to announce the next seminar?